think I have to share my presentation yet. Yes. So yesterday we have discussed about target identification, modeling, and validation. Okay. So we have seen. I just didn't showed you how to use the softwares, but I told you what are the softwares we use for target modeling, target identification, and target validation, isn't it? So first, I start with target modeling. Oh, still the screen is yet to be shared. Yes. So can someone tell me what are the softwares we can use for target modeling? Softwares used for target modeling. Target, I mean protein receptor. Very good. Swiss modeler. Swiss modeler uses what method? It uses a method called homology modeling, isn't it? Likewise, what are the other tools we have discussed other than homology modeling? So we also seen the second method as threading. Very good. For threading, the example is eye taser. Your answers are right. And what is the third method of modeling the proteins? The third method is very good. The answer is ab initio. Can I get at least one software for ab initio? I have shown plenty of softwares from Xpassi website. In Xpassi, for all the three, we have I think in the ABA. Okay, so I've shown you the page. Now again, I can show you once again the page where you found. So actually, it is from the xpassi.ch. Okay, so in that website, okay, you can find the three methods of protein structure modeling. For these three methods, you have plenty of software tools available. If you click the hyperlinks, it directly take you to the homepage of that particular websites. Okay. So for having issue, you can remember Anolia. Of course, it's there in the Swiss modeler thing, and the project. Project is available even separately in a separate web page, and also I think earlier we had embedded with the Swiss modeler. What if is of course a separate kind of website where you can check the binding sites, and even we can check mutations. We can also check the structure verification. So all these things we can able to do in ab initio. Okay, so these are some of the tools we have seen for target modeling. Very good. The answer is Rosetta. So if this is for target modeling, how to validate the target binding sites? And before going to the target binding sites, how to validate your protein model is good or not? If you model a protein, you have to validate, like you have to tell the quality of a protein is good, isn't it? So, what are the tools we majorly discuss to check the quality of a protein? Even now, I just showed you a kind of a tool called What If. This What If can also tell you how to the protein structure analysis program for mutation prediction, structure verification. But I told you one more tool on yesterday. I told a tool meant for assessing the stereochemical quality of a protein. A famous Indian scientist has come up with a kind of a plot which can be used to find the secondary structure of a protein. Actually, if you see this structure, we have very good. The answer is Ramachandran. The Ramachandran plot is used to assess the quality of a protein. Okay. If you model the protein, immediately you go for checking the Ramachandran plot. The Ramachandran plot is available in almost all the commercial softwares, the computer-aided drug design softwares. For example, if you see the Discovery Studio, which is a package and a product of BioVia, you have a Ramachandran plot inside. Even in Scroidinger, if you see even Triposibel, in almost all the softwares you have the Ramachandran. But for the free ware usage, you better go for Swiss model. Okay. So to validate the protein, there are plenty of methods to validate the protein, but the most common which can be available in internet to validate is this Ramachandran plot. Even if you predict the model of a protein, okay, where the structure is not known, also have to refine the protein model. Okay, so there are so many concepts involved in modeling, not just modeling using any of the methods will give you a kind of a good quality of a protein for docking or simulation. So even while during the modeling, you can find some of the protein amino acid residues are missing. If so, you have to add the missing residues. Then only you can go for docking as well as the simulation. Especially for simulation, the quality should be 100% perfect. If the even during the X-ray or NMR crystallography, the NMR studies or X-ray crystallography studies, sometimes the protein structure won't be good. That you have to refine it using uh, the tools available in uh, the commercial softwares like Discovery Studio. Now I try to open a tool called ASD Chem Sketch. Yesterday, I can't able to show much illustration, isn't it? I just explained you with a few kind of a tools. Now I'm going to illustrate with you how to use these kind of a softwares. First, I think I can take you to the ASD Chem Sketch. So if ASD Chem Sketch, if you default type in Google, okay, you'll be getting like this. Okay, so ChemSketch is the tool, and ACD, of course, is the name of a company. Okay, so after installation of this ACD ChemSketch, you get a window like this. Now, I tell you, I mean, I'll be explaining you how to get a 3D structure of a small compound. Like if you want to design a drug, but generally you design a drug if the structure is not known, if the structure is not, I mean, if the structure is not available in database, but if the structure is known, you can download the 3D coordinates from the NCBA PubChem, isn't it? But okay, for example, I'll be explaining you how to design a drug using this tool. Okay, this is a Kim Informatics tool. So initially, I like to give you with a small example. So now I'm going to use an example like aspirin, how to draw the molecule aspirin using ACD Chem Sketch, Chem Informatics software tool. So actually, what is aspirin? It is acetyl salicylic acid, isn't it? Acetyl salicylic acid. You have carboxyl group in one end, and in another end you have. So if you want to pick hex, the benzene ring, you can choose this. Okay, and so also you can find other rings. Okay, six-member ring and five-member ring. If you want to choose, you can choose these functional groups present here. And also we have so many kind of other functional groups. Okay, for the molecules you have. For the atoms, you can pick from here. Okay, almost all the atoms in the table you can able to pick from here. Okay, so now I'm going to draw a simple compound called aspirin, which is a pain reliever. Okay, and I determine some kind of a properties, and I take you how to further go with this as ASD chem sketch. Now, the structure of acetyl salicylic acid, I can show you first initially the structure of acetyl salicylic acid. Then I can go to drawing this structure. So, the aspirin structure. So, if you see the structure, okay, the simple structure, if you want to remember, you can remember any of this. Okay, so like, can we go for this kind of structure? Okay, so I think I can take this from this structure. Okay, so initially what you have an alkyl group and you have double bond OH. So this I'm going to draw now. So for this, for picking the alkyl group, pick this. So now I just click here. You got a kind of an alkyl. So here I'm going to branch and branch one more. So here you have double bond. So for having a double bond, click here. You got double bond now. And here you should have O, isn't it? So if you click O, it will be converted as O. Here you want to have OH. Just because I have a single bond, if already the O is selected, if I click here, you got the OH. So this is COOH, carboxyl group. So the other functional group is here. Okay, If this is ortho, this is a kind of meta, I think so. So if you take here, I think ortho, meta, para. So however it is, okay. If there is one more functional group here, okay. So I'm going to draw this, sorry. Already the O is there. So if you want to revert back, okay, control Z, okay. The oxygen should be deselected. So it's not deselected. So better select the carbon. Okay, if you select the carbon, so if you want to draw a alkyl group, if you click here, you get an alkyl group. So now see the structure. What you have to get, this structure is okay now. Now we have to get O and an alkyl group and double bond O, isn't it? So here you should have O initially. So I fix the O and then I have to go to the carbon because you should have, no, sorry. Actually, again, I have to click oxygen. So just because if I click here, it is automatically converting as CH3. So what I'm going to do here is I'm going to click the C and you want to draw an alkyl group like this, isn't it? So I'm going to stretch this. If I stretch this, I get a kind of alkyl group. Okay, I got this. Now I have to stretch here. So I got like this also. Now stretch the bond here, go for double bond, and here you should have OH. So we got this structure. Okay, so check the structure. So this is how the structure looks like. Okay, so this is a structure of aspirin, that is acetyl salicylic acid, which is a pain reliever. So whatever I have drawn, okay, in this software, okay, if you want to know what exact compound it is, think we already know, I already told you this is an aspirin and I just drawn. But if you have a structure, okay, an inactive, and this inactive can be used for treatment of any specific disease in your research. Better draw the compound and then you check whether this compound is available in PubChem or not. Initially, now I'm going to close this PubChem window. I'm not having any window at present. Now I'm going to click here in PubChem. So it is going to ask you whether you are going to search exact structure or not, whether you want exact structure or you want a derivative compound of aspirin. If you want to search substructures, it won't you won't get the same structure, but you get the similar structures of that. Okay, but if you want exact structure, initially you check with it. Okay, so click OK. Now it is connecting the server. So whatever software we install, okay, whatever we are working in offline, it is directly connecting in online now. See here, it is connected to the website NCBA PubChem. You got immediately like aspirin. You got the result like aspirin. So not only that, yeah, you can connect this kind of a software, ChemSketch software with PubChem. You can also connect with e-molecules. E-molecules is a database where you can use even for purchasing the chemical compounds from the companies. This is a vendor based. Okay, if you want to purchase, even you can purchase from any of these chemical companies. This aspirin is manufactured by so many companies, isn't it? So even the catalog number is mentioned. So it will be easier for you to purchase the kind of a compound. So the people, those who are involved in combinatorial chemistry labs, medicinal chemistry labs, develop few compounds, heterocyclic compounds. Draw the heterocyclic compounds, get the kind of a structure, same structure or similar structures, and try to check with the even E molecules to purchase that. Okay, and this is Chem Spider. Okay, this is another website. Okay, for a chemical molecule database. In Chem Spider, you get for the for natural plant compounds as well as for the drugs. If you want to get some properties information and more annotation on that, including the biological information, when does, so more informations are available here. Okay, so additional properties are also available here. So not only can limit up to NCP PubChem, even ChemSpider is, of course, a large database for annotating the small molecules. Okay, so likewise, okay, we have software not only for drawing this kind of, I mean, you can draw the chemical compounds and you can also get the 3D structure from that. Okay, so to get the kind of a structure, okay, so whatever structure I have drawn is a 2D. If you want to get this information in 1D, okay, you can use the same software to get smiles format. To get this structure in the form of a smiles format, okay, so you can check an option here. This is actually INCHI. This is for what? Smiles notation. So this icons is for smiles notation. Click here. You got a smiles form. So these compounds 1D information is provided like this. For a protein, how you find primary structure as the amino acid sequence? Likewise, for this 2D structure, this is the 1D information. Okay, this is the canonical smiles format. So if you want to get a 3D structure of this chemical compound, you can able to get it from MolSoft, Mol Inspiration. Plenty of tools are available. It asks you to just input the files format. So simply copy this format and paste it to get the 3D structure. The 3D structure you can download in the form of STF coordinates, PDB coordinates, .mol2 for coordinates. There are different formats for downloading the 3D structure of chemical compounds. Okay, good. Now, let me take you to these kind of a softwares also. So initially, I told you something called Mall Inspiration, isn't it? So you can go to any Mall Soft, Mall Inspiration, Mall. Plenty of tools are available. Mall Inspiration Galaxy 3D Structure Generator. If you click here, it asks you to input the smiles format. Now I'm going to copy this. Sorry. So already I think we have selected some more. Okay, if I want to copy this, okay, so I got the smiles, isn't it? So I have to come outside. It's not there. So I have to select this. Control this. Can't even deselect. So we have to copy and paste. Okay. Yeah, actually the option is there. I have to check an option. But you can copy this smiles format. Okay. If you know the smiles format, you paste it here. I think some kind of example is provided already. I take an example, I paste it here, okay, generate 3D. So we get a kind of a 3D information. Even you can get the information from here. And also we have a few more options available other than 1D formats. Okay, we can also go for 3D viewer, but I think for this you need a kind of this is actually a freeware, um, but only few options are provided to work on. Okay, if you want to use almost all the features, you should of course should have a kind of a commercial version. Okay, now let me take you to the peptide design. So for peptide design, we have a server called pepfold3. Okay, so if you type pepfold3, it directly takes you to this first URL, pepfold peptide protein structure prediction. So here you have to click run pepfold3. If you have amino acid sequence, okay, for an epitope, and if you want to predict it, so you can able to uh, paste the amino acid sequence. You know, the amino acid sequence are of 20 alphabets, isn't it? The 20 amino acids. So you can type randomly. Okay, so type the random amino acids. Okay, so we don't have P, Q, or S. Okay, I've typed some kind of amino acids. L stands for leucine, K for lysine. What is D? Aspartic acid. M for methionine. N for aspargine, proline. Okay, so likewise we have glutamine, arginine, serine, threonine. Okay, so the single letter amino acids I have pasted here. And now, if I want to predict the 3D structure, simply click run. It takes some time to generate the result. So before that, we have to input the captcha. So I'm going to type the captcha for the pepfold3. So presently the server is running. So within few minutes we'll be getting the result. Meanwhile, I can take you to the another software called Swiss Modeler for predicting the protein. And after predicting the protein, I tell you how to assess the quality of a protein. So we have to click Start Modeling, and this is the page of Swiss Modeler to work on predicting the structure for a sequence where the structure is not known. If you want to download a sequence, okay, better go to NCBI. We have plenty of sequences, more than 300 million nucleotide sequences and more than 40 million protein sequences are present in NCBA. But we have only 1 point some 6 or 7 lakh structures in PDB. So we don't have structures for all the sequences present in NCBA. And of course, this is the largest database for the protein sequences. So if you want to download any kind of a protein sequence information for which the structure has to be predicted, better use NCBA. But UniProt, you get only one protein information for a particular species. Okay, but here you get a plenty. So even from India, from Kerala, one scientist can able to post a same protein structure, and even from Tamil Nadu, and another scientist can post the same structure. Everything is possible here. The GenBank, the in NCBI, both for Bankit and Sequent tools for protein sequence submission can accept sequences from any of these scientists. But in UniProt, it is not like that. If you already have a structure, they won't allow you to deposit these structures. Okay, generally that is possible only with the NCB. Okay, so depositing the sequences and the structures. Now let me take you to the protein information. So first, if you want to predict a structure for insulin, of course, this is available, or we can give some protein for which we don't have a structure. So can you do you know any kind of I'd better take my protein of interest, TRAP1. So TRAP1 means tumor necrosis factor associated protein 1. Okay. So for example, I'm going to take even a species which is not known, at least for human you may have, but this kind of a species I don't think you have a structure, but it is a very large protein, some not seven amino acid. If you want to predict the structure for this protein, either copy the amino acid sequence, or you even can copy the entire FASTA format. However, you copy and if you paste it, only the amino acid sequence alone will be taken. So the FASTA format is converted to only amino acid sequence. Okay. So if you want to predict a structure, okay, directly if you want to get a result, click build model. Or else, if you want to select a template based on your requirement, first click the search template. Then you can, after getting the templates, you select a desired template, and then you have to click build model. Likewise, you have to do. And you also have options for predicting the to get the result in email. So initially, I'm going to give some kind of a title, and I'm going to give my email ID to get the model. So just like that, I pasted the sequence. I've given a name for the project, and then now I click build model. So meanwhile, I can check whether you got the result for pepfold 3, for which the peptide structure has to be predicted. Yes, it's still running. Okay, we are yet to receive the result. After receiving the result, I can show you. Okay, so I think this server will take a minimum 5 minutes, and this server will take minimum of 10 minutes to predict the results. So I am going to take you to today's session. Okay. So whatever I explained you or something is related to the yesterday's session. Okay. Some hands-on experience I've shown. Now let me take you to this today's chapter on essentials of drug design. If you want to predict drug, okay, whether the drug for a particular disease can be even for it's not exactly SARS-CoV-2 for any diseases. Okay, so what are the different computational methods in drug design? So if you see this, we have nearly 10 to 12 methods, isn't it? So what are the most important methods we generally use? Initially, we can go for drug likeness prediction through these tools and ADMET prediction. I mean, absorption, distribution, metabolism, excretion, and toxicity. So even before ADMET, we can also add L. L stands for liberation. So LATMET. Okay, so these parameters are pharmacokinetic parameters. If you want to determine, okay, the properties of the distribution and toxicity, all parameters, better we can go for this kind of online tools like Swiss ADME or pre-admit. So if you want to predict the activity of a compound, that is, if you see this ADMET, QACR, this comes under ligand-based drug design. Okay. So even drug likeness prediction comes under ligand-based drug design. That is, if you have a drug, that drug information alone is sufficient, okay, to predict these properties. But if you see the next methods like talking, molecular dynamic simulation, here you should have a 3D structure of a drug as well as 3D structure of the receptor. If only if you have the 3D structure for the small molecules and macromolecules, then only you can able to check whether your drug design is good or not. Okay, to validate the drug design, you should have the 3D structure for both the ligand as well as receptor. There is also one more method of drug design that is hybrid based. Okay, these if these three are ligand based and docking and simulation is the receptor based. If I tell receptor based, you also include the ligand inside. Okay, and there is another method called hybrid based. Example is pharmacophore modeling or pharmacophore mapping. Okay, so we can't tell that you need both the ligand and receptor. Sometimes even the ligand alone gives the information. So that's what we call that as a hybrid based kind of drug design. Okay, so these are generally the tools used by most of the scientists. Even if you want to go for uh, the inverse talking or reverse talking, that is possible with a tool called ACID, and few more tools are available. That is generally what is talking. Actually, if you want to check the binding affinity of a ligand with the receptor, we go for the docking. There is a concept called inverse docking, which is just quite opposite to that. So we have everything in the subsequent slides. When I try to explain, I think you can understand all this. Okay. So I just have a view. So these are the computational methods of drug design for any diseases. Okay. Now let me take you to each and every method in subsequent slides. Okay, number one, I'm taking the case here. 1 minute. Let me go through some slides of. Okay, so whatever is uncovered, I'll be explaining on tomorrow. So first, I'm taking this QS here. The QS here is a quantitative structure activity relationship model. So here we can able to predict the activity of the compounds. Okay, especially if you are a microbiologist. Okay, generally the activity that we calculate through MICs, minimum inhibitory concentrations. Negative logarithm of MIC is what we call it as activity. So if you determine the MICs for different compounds, the activities are automatically calculated from the mathematical formula. So whatever you calculate activity for the known drugs, okay, and if you have a drug for which the activity is not known, that can be predicted from the already predicted activities of the drugs. Okay, I mean, first you have to build a training data set and you are going to build this linear regression. Okay, and whatever model linear regression model you have developed, if you want to predict the activity for the unknown compound, you can able to predict from that. Okay, the test data set can be used to check the activity from the training data set. So this is like a machine learning approach, of course. And if you want to use that, okay, predict the activity, so this kind of a schema can be used. So initially, you should have a structure for the compound, and then for the structure of the compound, you should also have some molecular descriptors. The molecular descriptors here, I mean, everything hydrophobic, hydrophilic. You should have electrostatic descriptors. So many physical and chemical parameters related to the functional groups of the ligand. So if you take a drug, there are so many functional groups, isn't it? So we observe all the electrostatic descriptors, especially in the molecular descriptors. And then if you see from the already existing compounds, you predict the activity, isn't it? So you're going to build a training data set model. So that can be done, and the unknown compound activity can be predicted like this. So this is the schema we can follow, and these are the softwares available. And if you see the like just like activity, even the other properties of chemical compounds can be determined through QSPR, and the reactivity can be determined through QSRR. And if you have a series of compounds from a same plant, okay, then that can be determined through QSCRS, because if you take some group of compounds can be in alkaloids, a similar group of alkaloids have almost same molecular weight. If you have same molecular weight, so if it is very difficult for you to predict these kind of relations, you can build a relations to the chromatography, the QSCS, and you can able to predict it. Likewise, you have toxicity relationships, electrochemistry relationships, and biodegradability relationships. Reverse docking. So I think first I have to go to docking. Then if I go for reverse docking, you can understand. Anyhow, the docking I'll be taking you in a separate session. Okay, so now I just tell you what is docking and what is reverse docking. First docking. So the docking, if I want to check the binding affinity of a ligand with a receptor, okay, that is a drug with a disease protein. So you will be checking the binding affinity by binding with it, and you determine through energy score. Generally, if you want to practice talking, it is feasible with you to practice with Autodoc. The Autodoc is the excellent tool for getting results with respect to energy. Even you can check the energy, and even if you take this result from Autodoc, it will be easier for you to use that as an input for simulation. So generally, we work docking with Autodoc, and we take the output and take that as an input for Chromax for doing molecular dynamics simulation. So then if you ask me what is the difference between docking and simulation, docking is just checking the binding affinity of ligand, that is drug with the receptor. This is protein. But if you see the MD simulation, you are going to check not only the binding affinity of the ligand with the receptor, but we are going to check with respect to time. Okay, so like if you take a drug, okay, we have to know how much drug protein binding has happened. Okay, so the drug protein binding, okay, is up to a particular time. We have a concept called residence time. Okay, if you take a paracetamol, we take 3 doses per day. It means that the residence time of paracetamol in our body is just 8 hours. But few drugs, I think even if clinicians prescribe, they prescribe to take only one drug, that is on night. So before sleep, they will be asking you to take a drug. If so, the residence time of that particular drug is 24 hours. Okay. Some drugs are prescribed for 3 hours, then it is 8 hours. Some drugs are prescribed for two doses, then it is for 12 hours. So it varies, isn't it? So one should have a knowledge on the binding of drug with our protein, okay, with respect to time. So this we can able to practically work through MD simulation. So MD simulation also on yesterday I told you that can also be used to predict the 3D structure of a protein from the X-ray coordinates. Okay, that is one application. This is a second application. If you want to check even the binding affinity with respect to time, okay, you can also use the same MD simulation. MD simulation is a common technique used for various purposes of drug design. Okay. Now, if you see this kind of reverse docking, so there, how I mentioned, you should have a 3D structure of a ligand and receptor to check the binding affinity, go for docking. But whereas in reverse docking, okay, you take a single ligand, okay, you're not going to bind with a single receptor, but you bind with plenty of receptors. So this example you can able to find in checking the nature of your drug. Like if you are taking a drug, for example, if you take aspirin, aspirin is pain reliever. Everyone can accept. But this is not good for heart. It gives a cardiac problems. Aspirin generally some 20 years before everyone if they face uh, body pain, so everyone can go for prescribing the aspirin. But it resulted in so many cardiac failure problems. If just because of that, it's a banned drug, of course, nowadays. So if you see this, okay, the effect of aspirin, the aspirin is very good on binding with the acetylcholine esterase receptors. Okay, so that is COX, that is a COX inhibitor, isn't it? So it better binds with the COX inhibitor, I mean COX enzyme, but it can also bind with the heart receptors. Okay, so if it binds with the proteins and if it inhibits the heart function, okay, then obviously it results in failure of heart. Likewise, okay, so if you want to check even the side effects of a drug, not only the main effects of a drug, better go for reverse talk. Then you can able to check, okay. So we have a websites for doing the reverse talk. Simply they ask you to input a single 1D coordinate, that is canonical smiles format. Or even if they ask you to input it to a 3D coordinate, you input that. Generally, they ask you the canonical smiles. Most of the websites ask like that. Simply input that, and then you can check the binding affinity with respect to plenty of receptors. Here, but in docking, what you do, you take multiple compounds, you dock with a single protein target, and you come up with which is the best drug. If you're doing a kind of a drug design, you're not going to take a single drug and bind with the receptor. According to your research, if you design, if you go for a schema, you take plenty of compounds and you do the virtual screening. Okay, the virtual screening can be done by binding of each and every compound with the receptor, come up with a best drug. So that we call it as a lead. I think I explained you the same thing on yesterday. But here you take a single ligand, bind with the multiple receptors. Reverse stocking can be done with acid. This is the software tool and this is the URL. Now I found this server is down. We can't able to work with it. Might be after 1 month we may get fine. This server is active. But you can also practice with another software called online tool called reversestock.biology. So this is the URL. Actually, if you simply in Google, if you type reverse stalking, okay, and if you got like if you get like reverse biology, better use that link and you can input a kind of a ligand and you try with so even the example tutorial, everything is provided in this tool. Now, molecular modeling. Yesterday, even today, I'm explaining about this. Of course, an important method, isn't it? If only if you have a 3D structure of a ligand, 3D structure of a protein. Okay, then only you can able to go for checking the binding affinity through docking as well as checking with respect to time, the binding affinity with respect to time through empty simulation. So this is of course a most important step. If you want to model the proteins, go for homology modeling, threading, or ab initio modeling. And these are some of the tools available. So now can we go to the output? Okay, we can check the output now. So we got an output for the protein, but before that we worked on peptide, isn't it? Whether we got a model, missing services can be found here. Okay, we got the model. See here, I'm just tracking down. This is my peptide. This is my peptide. So methionine in fourth position. Can you see this? Here you can find the whatever I select, you can find that methionine 4, aspargine fine. Even you can get different models if you want to see ball and sticks. Okay, if you want to get a lines, you want to color by the chain. color by, okay, so whatever you like, okay. So if I select, even you can get what amino acid exactly is that. Okay, not only amino acid, also I get the atom position. So leucine one, carboxylic acid, Ca, no, Ca meant for carbon alpha. This is carbon beta. Cb means C, carbon beta. So likewise, you can find the atoms and amino acid positions. If you want to download the PDB structure of your peptide, okay, so track down, check here. You have to get like this model1.pdb. Okay, so here you have an icon. Click this. You got a coordinates like this. So you get only the coordinates, the 3D coordinates. You have to copy it. I suggest you to use WordPad for saving this format. The 3D coordinates of protein or peptide to be saved in editor, you choose a default editor as WordPad. Okay. Yeah, but of course, Notepad is also not bad. Okay. Let me, even if you go to the WordPad, ensure you have to keep it in text doc. But in Notepad, defaultly, it is in text doc. Okay. So if it is an open document text, I think you may get in trouble. Better go for text document. So I think it's even better to go with Notepad. Okay, so I just selected text document. I'm going to give but before that, I'm going to save it in desktop. So this is a peptide model. So I'm going to name the file as peptide underscore model dot pdb dot pdb. Whatever I'm going to mention, the file name dot pdb should be in double quotes. If you are using two names like peptide and model, better use underscore. Don't use space. That won't give good results if you go for docking or simulation. You get in trouble. If you are using two names, better use underscore. And whatever extension you are going to give, the PDB is the default format, not only for the proteins, but this is also a format for the peptides, small compounds, etc. Okay. So PDB is, even though the name is protein data bank, that is a misnomer. Structure can be used for all the molecules. Okay, this is the format for the structure of all the molecules. Now I'm just going to click save. Yes, I just saved it. If I want to open that peptide underscore model, so sort by date modified. I have this peptide underscore model. I've already installed BioVia Discovery Studio 2023. So this is the free visualizer to visualize compounds, peptides, protein structures. Almost all the biomolecules can be visualized using the free visualizer. I think we have the PDB file must contain at least one header. Yeah, actually there is some problem with the header. Protein structure, this is simulation box. So header is here, but it's that down. Okay, I can check what else can be corrected. It started with remark. The header is generally should be in the first line. Okay. Let me check whether you get answer or not. I just saved it. Going to try once again. But that can be visualized. This can be visualized in other. I have Python molecular here. If not in Discovery Studio, I can check it here. I got a structure here. Okay. So there is some problem with the Discovery Studio visualizer, but I found the structure in the Python molecular view. Okay, can also choose accordingly. Okay, so we have to correct the coordinates according to Discovery Studio visualizer. Okay, however it is, okay, we have seen the 3D structure of the peptide. Okay, now let me go to the predicted model, protein model. So actually, The Swiss model is going to generate two base models, model 1 and model 2. So you have to choose the base model based on sequence identity. Okay, that gives you more accurate information. Okay, so sequence identity, if you see here, this is 77.61. I think model 2 has covered most, 89.19. So it means that we don't have a structure in database. It is sure. Okay, that's because we don't have a structure for the protein, whatever I typed for a particular species. Okay, I just use this project for predicting. Now, whatever structure you have downloaded, I mean, you have, if you want to download, so you have to click the, uh, so where is this? So you can able to download this structure by click download files, PDB format. Okay, so I'm simply going to click PDB format, going to save somewhere in desktop, model underscore 2 is my protein. Okay, so I have downloaded that. If I want to open the model underscore 2, open with Discovery Studio. Now the protein structure is displayed. Okay. Now whatever structure we see, okay, better go. If you want to see the coordinates and some information of the protein, click View Hierarchy. If you click this View Hierarchy, you got a chain, and these are the amino acids of the protein. 707 amino acids are shown. So luckily, we can able to find 1 to 707 amino acids. It means that you don't have any missing residues. Okay, so just because you don't have any missing residues, this will be uh, easier for you for going with proceeding with docking as well as simulation. If you have missing residues, you have to use a protocol to add the missing residues. That is possible with Discovery Studio Visualize. If you want to add the terminal residues, if any of the initial residues are missing or final residues are missing, that is possible. Generally, the initial residues are easier to add. Okay, let me complete it. So to visualize this protein molecule, okay, you'll be using the Discovery Studio Visualizer. You can use plenty of options, okay, which are very easier for you to take it in the form of a publications. Okay, so I converted in the form of background file, and now I have this alpha helices in red color and beta strands in the kind of a blue color. If you want to zoom in, zoom out. If you want to, if I just type few amino acids, I just selecting few amino acids by clicking Control. You can also see these amino acids separately. I think just whatever selected is there in this region, this loop region. Okay, whatever is selected, I think that is here in this loop region. If you want to see this amino acids in a different state, okay, in the form of a ball and stick, I click here in ball and stick. See, only these amino acids alone now shown in ball and stick. Okay, so scaled ball and stick, so it will be on automatically converted like this. So not only you can able to have a structure, this is a solid ribbon structure for a protein. If you want to select specific amino acids and want to convert stick model or ball and stick model, you can also able to convert. So this is useful when you predict the binding sites, and if you want to have a binding sites in a different shape, okay, you can show separately like this. You can see the entire protein. You can also see this kind of any selected binding sites. And whatever you find here, this is the loop in the 3D stage. Okay, now we have seen how to model the protein using Swiss modeler. Of course, this is a part of a drug design, and how to visualize it. Now, if you want to assess the quality of a protein, so we can click structure assessment. You know, the structure assessment take you directly to assess the stereochemical quality of a protein using Ramachandran plot. Then how to validate the structure quality? So the structure quality can be determined, okay, by checking the location of amino acid residues. If you see the protein, whatever I have taken is 707 amino acid in length. So all the 707 amino acid, if you select this, okay, this is one amino acid, this is another amino acid. Okay, so we have so many amino acids, isn't it? Whatever, even if you select, okay, if you see here, even you can get what amino acid we have selected. So simply I click here and I can find it. So this is the amino acid. And if I click here, so this is the amino acid location you see. That is iso, what is that? Lysine 547 position. So whatever I selected is lysine 547. Okay. So all these amino acids should lie only in this dark green region. If it is lying in this region, this is most favorable. But allowable regions are in this light green region. So if amino acids are lying in this light green region, still you can tell it is allowed. But if you find these amino acids, these are not allowed, especially for few proteins, even the amino acids or lies somewhere here, somewhere here, somewhere here, somewhere here. So if the amino acids are like that, okay, the protein quality is not good because the amino acids are not in the dark green as well as in the light green plot. If you see here, whatever structure I have predicted from the 89.97 similarity, okay, so I got Ramachandran favored as 95.46. Very good. When I can tell term tests are very good, if you get 80% and above, you can take this protein structure, whatever you have predicted and downloaded for docking and simulation. So the Ramachandran favored residues should be 80% and above for research. But just for that, for its sake, if you want to work, at least the proteins Ramachandran structure, whatever you predicted, the Ramachandran favorite should be above 50 or 51 or 51. Okay, even if it is above 51, you can take it, but I can't able to guarantee whatever you predict is precise or not. But if it is above 80, good. If it is above 90, very good. If it is below 50, don't take the structure for any computational studies. Okay, it gives you a kind of a false positive or false negative results. Okay, so you have to check even the quality of your protein through assessing through stereochemical quality of a protein through Ramachandran plot, which is available in Swiss Modeler itself. I'm not going to any other website. From XPC web server, you have a tool called Swiss Model online tool. From that itself, have done all this. Okay. Generally, the allowed residues speak about the glycine and proline residues. Okay. So even you have that information. Okay. The glycine, which is the most flexible, and proline, which is most restricted rotation amino acid. So these two are used for generally checking the quality, stereochemical quality. So this is the principal line behind. Okay, let me take you to the presentation. Now, we have seen even how to model the protein, how to assess the quality. Okay, now take you to the molecular docking. Of course, we have a separate session for molecular docking. I told you just the binding affinity, checking the binding affinity of the ligand and receptor is what molecular docking. So for this, you have an algorithm and scoring function. More about this we discuss while going for a practical demonstration with Autodoc. There are also a few more tools, the commercial software tools available for docking. So in Autodoc, you get only one score, we get like a binding energy score. But whereas here, you get plenty of score models based on the physicochemical property of drugs as well as based on the interactions of ligand with receptor. So this kind of calculation, that is a total potential energy, binding energy calculation, is commonly observed in both docking, dynamic simulation, etc. So the sum of bonded interaction and non-bonded interaction is what the total interaction, and from the interaction you can able to calculate the U, that is potential energy. That is potential bonded energy is the sum of potential bond stretch, bond bend, and rotate along bond. So this is bond stretch, bond bend, and rotate along bond. Okay, and we also have a non-bonded potential energy, which is calculated from the weak forces. van der Waals and electrostatic, and the sum of bonded and non-bonded is what? The total uh, potential energy, okay, binding energy. So this is the common concept, and I told you what is MD simulation, how you distinguish with docking. So here we are also concerning on time, okay. And if you see the softwares, okay, these are the softwares available. Generally, the Chromax is the freeware, which can be worked only on Linux platform. So I think nowadays we also have a kind of interface, okay, to work on Windows also. Okay. And if you see some kind of outputs like RMSD, RMSFA, these are some of the simulation outputs. This will be explained in more during a separate session on MD simulation. Now let me take you to the expression profiling. This is another kind of advancement in drug design. So if you want to go for precision medicine design, don't just take a single drug and single receptor. You should take multiple drugs. Okay, you should take the drugs and you have to screen the best drug for a particular receptor. Okay, whatever you do, we call it as a molecular docking. That won't give a precise drug for a patient. If you have patients for a cancer, you have to tailor the drugs. I mentioned on yesterday, you remember. So how this can be done? This can be done if you use system biology approach. In system biology approach, you will be checking the most important protein and what are the interconnecting protein. For example, if GNAI3, if this is my important protein, and if you target a drug with this protein, I can't tell. I give you the successful result. You should also target the binding proteins of that hub protein. So you should also talk with MapK3, CACTV. So you have to do with all this. So you can't able to even go for reverse docking, okay, by docking with all the receptors. If you are focusing on this is the important gene diagnosis for your cancer, and if you want to target this gene alone, no need to target these genes. Only you have to target the binding proteins of this but specific protein. You got it? So this kind of an expression transcriptomic data profiling has to be done. Okay, so you have network databases, you have softwares like Cytoscape, so you can screen it. Okay, you can enrich the gene and you can bring the top important genes. And from the top important genes, you can also use for a combination of drugs and you check which is the best one. So this kind of research happened in 2020. I published in Elsevier journal, okay, on ovarian cancer. Pharmacophore modeling. This is another method. Okay, in pharmacophore mapping, we generally go for taking a ligand. That is, if you take a ligand, we take a backbone as the important structure. We attach or detach the functional groups to get a desired biological activity. Okay, we fix a ligand structure, okay, backbone of a ligand structure, and then we attach or detach. So likewise, if you see this slide, okay, if the ligand looks like this, okay, and if I attach something like uh, the OH group, okay, and if the OH group gives a better binding affinity, then you check the possibility where you can attach more OH groups in the ligand. So based on that, you can able to check the desired activity. So the pharmacophore thing can be done through several steps. Pharmacophore modeling followed by the mapping. So generally, if you see pharmacophore, I already told it is an abstract description of the molecular features that are necessary for molecular recognition of a ligand by biological molecule. So if you take this ligand, if I attach a kind of a CH3 and followed by OH, that is CH2OH, and if this functional group gives a better kind of a binding affinity, okay, so all this thing can be predicted only through this form of code. So attaching and detaching the functional groups are automatically available in the software. Okay, you can synthesize as number of compounds with the backbone already fixed. Okay, so like if you want to have two rings and you have to have this attached ring to that, that is to the CH2. So you just fix this alone, and you can use the fragment-based drug screening. That's what we call it as a pharmacophore mapping or modeling. So for this, we have plenty of tools. So I basically use Jing Farmer. This is the Jing Farmer used for a particular platform. So everyone can also try this. Simply go to the Google and type the farm mapper, and you just give. You have to input a ligand in Smiles format. So just if you have a Smiles format ligand, you just paste it, and if you click enter, obviously get series of drug target receptors. You will be getting different target receptors, and you also get your drug is binding the best fit with what receptor. Okay, so this is best fit with DNA directed RNA polymerase. So whatever compound I used is the best for treatment of RNA viruses. That's what I got. This kind of okay. Likewise, you have uh, the farm mapper. Okay, this is another tool like Zinc Farmer. I think I already explained you about the QS here. Okay, so now I told you a few important methods of computer-aided drug design. And I think few more methods are available. So that methods will be continuing on the next session. So the next session, that is fourth session, is on the left-out methods of computer-aided drug design. So that is related to drug likeness prediction, admit properties prediction. All these things will be seen in the next class. Okay. If you have any doubts, you can ask me. No? So I've completed my third session on today. Can we use Pyrex for docking? Yes, definitely we can use it. This is used for visual screening, not docking a single ligand with a single receptor. We dock multiple ligands with a single receptor. Docking multiple drugs with a receptor can give you the best lead. Okay, you have to identify a lead. You have to optimize the lead. Then only you can go for any kind. Even optimize after optimizing the lead, you can extend uh, this concept with pharmacophore mapping or modeling. Now, after pharmacophore mapping or modeling, you can get a base structure from the lead, and then you can go for the docking as well as simulation. So initially, for lead identification, better use the kind of virtual screening software and get a best lead, that is a best truck, whatever you get. And now go for pharmacophore mapping, attach or detach groups, and then you get. So farm mapper is used to find the target of our ligand. Yes, of course, that is also used to find the target. This is a second application. The first application is to you have to go for a fragment based ligand. அதாவது whatever drugs existing in market, you can't able to claim that that is the best drug for a receptor. Okay, for that disease, you can also check whether you can able to improvise the drug. If instead of taking 3 doses, if you can able to take a derivative of a drug for 2 doses, it will be better, isn't it? So one should think about how to reduce the dosages. Okay, and whether is it targeting most of the population or not. Precise targeting is important. So even if the drug design is not completed, it's a continual process. So one can also able to improvise the drug for a disease. Got it. So of course you have to use the sequence of tools in computer drug design to come up with a best drug for a disease. For a disease. Okay. Any other doubts? Good. Thank you very much for patience, listening, and for a good interaction. So just because we are to show the hands-on experience, I didn't ask much of the questions.